The first place that food and drink enter would be the oral cavity inside the mouth. Inside the mouth, we have two different types of digestion. One would be mechanical digestion, which would be the result of the muscle contraction and also using our maxillary mandibular teeth, basically chewing. The mastication is the mechanical digestion happening inside the mouth. We're breaking down large piece of food into smaller pieces. And also we mix food, solid food, with saliva. So eventually we need to create a soft, flexible mass that is called bolus, and then swallow bolus. However, before I follow bolus from mouth into pharynx, let's clarify that inside the mouth, we have also release of saliva from salivary glands. We have 3 pairs of major salivary glands and many minor salivary glands. All of them secrete saliva into the oral cavity. Saliva has many different functions, which in a separate video I'm going to talk about the different aspects that saliva help us. But in saliva, we have a very important enzyme named salivary amylase. That's the one that inside the mouth starts breaking down a carbohydrate such as starch. So we say inside the mouth, the chemical digestion of carbohydrate is going to start. And when the time comes to swallow, obviously we use our tongue to push bolus, that flexible mass, toward our oropharynx. So tongue moves up and also moves backward and pushes bolus into oropharynx. And as soon as bolus gets inside the oropharynx, the receptors that we have in the wall of oropharynx fire signal to our swallowing center, deglutition center, which is located in medulla oblongata of brain and also the lower parts of pons. And from here, the involuntary stage of swallowing is going to start. So when we push bolus from mouth into oropharynx, obviously consciously, voluntarily, we can control that stage. But from oropharynx, the rest of the swallowing would be involuntary. Exactly when we have bolus inside oropharynx, we see several amazing things are going to happen. One is that from the neurons in swallowing centers, signals will be fired to the muscles that we have in soft palate. Soft palate moves up and closes nasopharynx. So we do not let bolus or drink to get inside nasopharynx. And then the contraction in the wall of oropharynx moves bolus down. And we know that this is the moment that epiglottis also has to close the larynx. In general, during swallowing, we close nasopharynx using soft palate and we close larynx using epiglottis. So breathing stops for that short period of time. And then from oropharynx, we move into laryngopharynx. And from laryngopharynx, we have to enter esophagus. Recall, esophagus sits behind larynx and trachea. At the beginning of esophagus, we have a sphincter. When we say sphincter, we mean some muscles that they have a circular pattern and they're in charge of opening or closing a pathway. We named the sphincter at the beginning of esophagus upper esophageal sphincter. It's made of skeletal muscles. When it opens, bolus from laryngopharynx enter esophagus, and then we have the coordinated contraction of the muscles that we have in the wall of esophagus to move bolus down. So let's follow bolus. It goes through this long tube. Recall that esophagus penetrates the diaphragm and eventually gets inside the abdominal cavity. Exactly at the end of esophagus, which is connected to stomach, we have another sphincter made of smooth muscles. We call this one inferior or lower esophageal sphincter, or in short, cardiac sphincter. So this sphincter opens and allows bolus from esophagus get into stomach, and then lower esophageal sphincter must close quickly. In those individuals that the lower esophageal sphincter fails to completely close after bolus enters the stomach, the gastric juice, which is very acidic from stomach, can go up into esophagus and start damaging the wall of the esophagus. So that would be the acid reflux that needs medication to make sure that the lower esophageal sphincter stay close after bolus gets inside the stomach. Inside stomach, we have gastric juice, which is very acidic. Its pH is about 1.2 to 3. And bolus, that is a soft, flexible, semi-solid mass, is going to turn into liquid. We name this liquid chyme. So from now on, I'm going to follow chyme that forms inside the stomach. Beginning of the stomach, the part that is connected to esophagus is called cardia. The upper part of stomach, exactly above my pen, that would be fundus of stomach. The middle part of stomach, which is a very large part, is the body of the stomach, and the end of stomach is called the pyloric portion. So cardia, fundus, body, and pyloric portion. I can see that inside stomach we have lots of these elevations that we refer to them as rugae. We can see them without needing a microscope. The small curve that stomach has faces the abdominal cavity. We name this lesser curvature, and we also have a greater curvature. And what is important is that this stomach that we see in the model is a stomach that has already received food and drink. An empty stomach is almost the size of a large sausage. One enzyme that works in the fundus of stomach would be the amylase that was released from the salivary glands. In general, salivary amylase is very sensitive to acidity, and it works only in that part of the stomach that its content is not mixed with hydrochloric acid yet. But as soon as the content that we have here get mixed with the gastric juice that is full of hydrochloric acid, salivary amylase becomes deactive and it stops breaking down starch. So we see that in a very small portion of stomach, digestion of carbohydrates continues. However, in the body of stomach, we have enzymes that they start breaking down protein and lipids. And eventually, when we have the digestion of these molecules, then it would be the time to empty chyme from stomach into small intestine. As you see, stomach is connected to the small intestine. The beginning of small intestine is called duodenum. Exactly at the end of stomach, where it is attached to small intestine, we have a sphincter made of smooth muscles. That sphincter is called pyloric sphincter. It is very important that when pyloric sphincter opens and we have contraction of the muscles that we have in the wall of stomach, we release a very small volume of chyme into small intestine. Simply because chyme that we have inside stomach is very acidic. Most enzymes that we have in stomach are okay with acidity. Actually, acidity helps them to do the chemical digestion. However, in the small intestine, the enzymes to do their job require an alkaline pH. So we open the pyloric sphincter, release about 3 milliliter of chyme here, and then we get help from the buffers that we receive and we have inside the small intestine to elevate the pH of chyme, bring the pH to a basic pH. Then that small volume of chyme moves down, and then we are ready to receive a small volume of chyme again. So every time pyloric sphincter opens, 3 milliliter of chyme empties into small intestine, and then pyloric sphincter closes. So chyme goes backward inside the stomach, and we wait for the next wave of muscle contraction to push chyme toward the pyloric sphincter, and then this sphincter opens to release again a small volume of chyme into duodenum of the small intestine. So what I do, I remove this part of the model, which is a section of the large intestine, to show you the inside of the duodenum. Because we see that when we are in the duodenum of small intestine, we have some circular folds. We call this pollica circularis or circular folds. They increase the surface area of the small intestine. Please keep in mind that in small intestine, we have to finalize the digestion and then absorb nutrients. So we have some digestion going on in the stomach, and also through the wall of stomach, we can absorb some nutrients, for example, electrolytes, water, small, short, lightweight fatty acids, some drug molecules. Let's say if you're taking a painkiller, obviously, you want in a short time the painkiller to be absorbed through the wall of your stomach and then relieve the pain. And also alcohol. So we have a limited absorption through the wall of stomach. However, most absorption happens in the small intestine. And as I mentioned, it is critical that when chyme from stomach gets into the small intestine, we bring the pH to a basic pH. So let's see some of the sources for the buffer that change the pH of chyme to an alkaline pH. First of all, I pay attention to liver. This is our largest gland, and as you can see, liver is divided into two lobes. The right lobe is larger, and the division of liver into two lobes is by this ligament that is called falciform ligament. Falciform ligament also helps liver to suspend inside the abdominal cavity. One thing that I don't see in the model, but it's good to note, is that exactly attached to the top part of liver, we have a very large ligament that is called coronary ligament. Coronary ligament suspends liver to diaphragm. So diaphragm actually is right here, and coronary ligament is the one that holds liver to diaphragm. The third ligament is this ligament that starts at the free surface of the falciform. And if you follow this ligament, you see that is attached to the umbilicus. We call this round ligament. Round ligament is the remaining of the umbilical veins that we used to have during the fetal life. So we have three ligaments here, and liver cells are extremely creative. They do many things, including forming bile. So that's liver that actually produces bile. And then we see that bile that we get from liver will be released into some pathways that we have for bile. So the bile that is produced by cells in the right lobe empty eventually into the right hepatic duct. The bile that is produced by the cells in the left lobe will be released into the left hepatic duct. And when right and left hepatic duct join together, the duct that they form would be common hepatic duct. When I follow the common hepatic duct, I see that is connected to the duct that is attached to gallbladder. Gallbladder is this tiny sac that sits below liver, and the job of gallbladder is concentrating bile and storing bile. This duct that is attached to gallbladder is called cystic duct. So here I can see common hepatic duct joins cystic duct, and what they form together would be common bile duct, which is a very long duct. It keeps going. The rest of it is behind the small intestine, and I can see here the rest of the common bile duct. So common bile duct starts right here as soon as cystic duct and common hepatic duct join together, and it goes all the way down here. And in this model, I see common bile duct joins to this long duct that we have in pancreas. We call this long duct either main pancreatic duct or duct of Wirsung. As soon as duct of Wirsung joins common bile duct, what they form together would be a dilated duct that opens into the duodenum of small intestine, and we call this dilated duct hepatopancreatic ampulla. So common bile duct and duct of Wirsung together form hepatopancreatic ampulla. Majority of population do have hepatopancreatic ampulla. It actually opens into the small intestine about 10 centimeters below pyloric sphincter. In bile and also in pancreatic juice, we have bicarbonate ion. So that's important that we release a very large amount of bicarbonate ion into the duodenum to elevate the pH of chyme. As you can see, about maybe 2.5 centimeters above hepatopancreatic ampulla, we can see this short pancreatic duct that is called either accessory pancreatic duct or duct of Santorini. So that would be another pathway for pancreatic juice to be released into small intestine. So as you notice, food and drink never get inside pancreas or liver. Instead, liver releases bile into small intestine, pancreas releases pancreatic juice into small intestine. In bile, we have, for example, bile salts that when they get into the small intestine, bile salts help emulsification. They're emulsifying agents. Emulsification simply means that bile salts help breaking down large globules of lipids into smaller ones. And also they participate in the absorption of lipid through the wall of the small intestine. From pancreas, we get pancreatic juice, which contains many different enzymes that can break down starch, enzymes that can break down protein, lipid, nucleic acids. And as I mentioned, the pancreatic juice gets inside the small intestine. So all the enzymes that pancreas releases will work and do the chemical digestion in the lumen of the small intestine. Speaking of the small intestine, the beginning of small intestine is duodenum, which is just about 25 centimeters. The middle part of small intestine is called jejunum. For example, here I have a closer view of the lumen of jejunum, and I can see that jejunum also contains those circular folds, plica circularis. Jejunum is just about 1 meter. The longest part of small intestine, which is called ileum, is the last part. Ileum is about 2 meters, and as you can see, when I follow ileum, I eventually see ileum attaches to the beginning of large intestine. So this is the end of ileum, and this is the beginning of large intestine, which is referred to as cecum. Exactly between ileum and cecum, right here, we have another sphincter made of smooth muscles named ileocecal sphincter. Clearly, when ileocecal sphincter opens, chyme from small intestine enters the large intestine. But before I get into the large intestine and go through its anatomy, let's recall that when we talked about the digestion of molecules, we mentioned that inside the mouth, digestion of a molecule such as starch starts, and then the next digestion, chemical digestion, happens in the stomach. For example, in the fundus, salivary amylase can continue breaking down starch. In the body of stomach, we have enzymes to start breaking down protein and lipid. And when we get into the small intestine, we try to finalize the digestion by receiving enzymes that we have from pancreas. Also, the cells that line the small intestine provide some other enzymes that we call them brush border enzymes. So we finalize the digestion here in the small intestine, and most absorption of nutrients into the blood happens through the wall of small intestine. And then finally, when we get into the large intestine, we need to note that in large intestine, we do not have enzyme. We have bacteria. We have a very large population of bacteria inside the large intestine, that whatever that hasn't been digested and absorbed in the previous sections of digestive system, some of those substances will be digested by the bacteria in large intestine. The beginning of large intestine, which is cecum, is attached to the long part of large intestine that is called colon. But before I walk you through the colon of the large intestine, let's pay attention that right here I have the opening of cecum into this coiled tube that is called vermiform appendix. This is a structure that does not really participate in digestion or absorption of substances. However, if there is any sort of obstruction in the vermiform appendix, inflammation happens as a result of it, which could be due to many different reasons. Then we have the inflammation of the appendix that is called appendicitis. And before appendix ruptures, definitely it has to be removed through a surgical procedure. So appendectomy is necessary. Now, from cecum, I start moving up through the first part of colon that is called ascending colon. And then right here, I see ascending colon has a turn, a curve to form the transverse colon. Exactly this part of colon, the curve, sits in this depression that we have below liver. Please keep in mind, in this model, they move the organs away from one another. Call this area hepatic flexure, and then the rest of colon that goes through transverse section would be referred to as transverse colon. And then exactly close to spleen, we have the next flexure, which is called splenic flexure. And then we start going down, that's descending colon. And eventually we get to part of large intestine that is called sigmoid colon. Please recall that when we look at the large intestine, we remember that ascending colon and descending colon are retroperitoneal. However, transverse colon and sigmoid colon are not retroperitoneal. And the next thing that we notice is that in the entire colon, we have bands of longitudinal muscles that they continuously contract. And we call this longitudinal muscles that contract continuously teniae coli. Their contraction helps this very long organ, because large intestine is about 1.5 meters in a living human, fits in this space. And due to the contraction of these longitudinal muscles, we end up with these pouches that we call them haustra. Now, when I follow the colon, eventually after sigmoid colon, I end up with rectum. And the last part of large intestine is anal canal, which is just about 2 to 3 centimeters. And anal canal has opening to the exterior, and the opening of anal canal to the exterior is guarded by 2 sphincters. The inner sphincter is called internal anal sphincter. That's made of smooth muscle. So autonomic nervous system controls the opening or closing of internal anal sphincter. However, we have the external anal sphincter that is made of skeletal muscle. So consciously, we learn how to control the external anal sphincter and control the process of defecation. We're speaking about an adult. So basically, to relax the external anal sphincter, we can rely on the cerebral cortex and how it communicates with the rest of nervous system to make the external anal sphincter muscles relax and open this sphincter.